Welcome to the Biological Life Sciences channel. Today we are going to see in details about Western blotting technique. So let's begin. Western blotting technique is a widely used technique in molecular biology and immunogenetics for the detection and analysis of protein. It is also called as immunoblotting because there is a use of antibodies in this, and it provides a qualitative as well as quantitative, semi-quantitative data for the different proteins of interest. Let's see the principle behind this. It relies on the specificity of binding of molecules of interest with a probe, that is primary antibody here. The prerequisite requirement for this Western blotting is SDS PAGE. That is, the electrophoresis used here is sodium didecyl sulfate polyacrylamide gel electrophoresis. Here the proteins are separated on the basis of size, shape and charge. A primary antibody solution is used here which binds to the specific protein. Then the secondary antibody solution binds to the primary antibody. The secondary antibody used here is a conjugated one, which is conjugated with HRP enzyme or alkaline phosphatase enzyme, and likewise the substrate is used so that the band can be visualized onto the membrane. Or you can use a radioactive detection using X-ray film or imaging system. Let's see the procedure in short. First of all, the sample preparation has to be done from the tissue from wherever you want to isolate the protein sample. The tissue has to be subjected for homogenization, then extraction of protein is done. It can be also sonicated. It is done at cold temperatures to prevent denaturation of proteins, as the proteins are very important here in this technique. Finally, observed concentration of protein and use spectrophotometer for protein concentration. The protein concentration is checked and then subjected for Western blot. Before Western blot, it has to be electrophoresed. The SDS PAGE gel is used here for electrophoresis. High percentage gel is used for low molecular weight protein and vice versa. The protein transfer is done onto a solid membrane here, which can be a nitrocellulose or a nylon membrane, onto which the proteins are blotted here. Proteins are negatively charged here in SDS PAGE because of the anionic detergent, that is sodium didecyl sulfate. The transfer can take place via two ways, either by capillary method, that is using the capillary action of the buffer inside a petri plate or a evaporating dish. It is kept overnight for the blotting of the bands. Otherwise, you can use a electro-transfer assembly where you use the charge to transfer the bands from the gel to the nylon membrane, and thereby electroblotting. Okay, the method is called electroblotting, where electric field is directed perpendicular to the surface of the gel, maintaining their relative position. This creates a sandwich where a fiber pad, that is sponge, is there, then filter paper is there, then blotting membrane, and then gel is there. Blotting membrane and gel are placed adjacent to each other so that the blotting of the bands takes place properly. So this process is called electrotransfer of bands. Generally, if you do a classical capillary action method, it takes about 12 to 24 hours, whereas while you do a electrotransfer method, it takes about 2 to 3 hours. After that, the protein is stained. The gel must be stained, as the proteins are not directly visible onto the gel. So dyes like Comasi brilliant blue or silver stain or deep purples are used here, and the gel is imaged using suitable instruments through which it can be permanently recorded. Onto the membrane where the detection is done, it is first of all blocked for non-specific binding of antibodies by using blocking agent. Then bovine serum albumin and non-fat dry milk are commonly used as a typical blocker here, so that the other antibodies will not bind other than the respective position of the protein band. It should possess a greater connection towards the membrane than the antibodies, so that it can bind firmly. Moreover, the membrane is also UV cross-linked and baked so that the bands of proteins are immobilized onto the membrane. And after that, the next step is antibody probing, where you use primary antibody which is poured onto the membrane, and it is kept in a moving condition for around 12 hours, where this primary antibody binds to the band on the gel, and then the washing is done. Okay, after the primary antibody treatment, secondary antibody or monoclonal antibody is added to this, which is linked to an enzyme that is used as an indicator for the location of the protein. After the binding of secondary antibody, washing is done using different buffers. Commonly there is TBS buffer or PBS buffer, which is used for washing purpose, and the washing is done to remove excessive secondary antibody so that it will not show us non-specific reactions while treating with substrate. Then protein is detected using two enzymes, as I told you, alkaline phosphatase AP and horse radish peroxidase, that is a perennial herb from which this enzyme is isolated. So respective substrate is used here. For HRP it's TMEH22, which forms a blue color. Four types of detection can be done. One is chromogenic detection, that is a colored substrate. Then chemiluminescence, means chemical creating a luminescence. Then fluorescent detection and radioactive detection can be done. Analysis and imaging. If you are using a radioactive detection, that is the primary antibody is a radioactive one, then it can be detected using X-ray film scanner or CCD machine. Benchmarking with marker proteins to estimate the molecular weight of the protein is also done in this case. And the verification can be done through qualitative and quantitative analysis to show the presence and absence of specific proteins of interest. This is the method of Western blotting in short at a glance. First of all, you have to pour the gel, then solidify the gel, then pipette out the samples and load it into the well. And the gel is run first using vertical gel electrophoresis. The protein is separated. Ideally, we do the separation in two bands. The one set of bands is used for blotting onto the membrane, and another set of bands is used to compare it after the electrophoresis, so that the position of the known band can be compared on the band as well as gel. So again, this is created into a sandwich where on one side there is a membrane and other side there is a gel, and it is sandwiched using sponge and filter paper soaked in buffer. It is then electrotransferred using classical capillary method or using electroblotting assembly. Then separate protein blot is done onto the membrane, and then it is treated with a shaker, incubatory shaker, where primary antibody solution is added to it, and wherever the band of interest is there, primary antibody goes and binds at that position, and subsequent washes removes the extra antibody from there, and then there is a treatment of secondary antibody. We have to make sure that the total surface of the membrane is removed with the primary antibody after before adding the secondary antibody, and then secondary antibody is added, and again wash is done, so that we confirm that neither of the vacant places has the secondary antibody. Then you have to add a chemiluminescent substrate, which will see, this is the secondary antibody linked to the enzyme. Now when you add a secondary substrate here, this enzyme reacts with that substrate and shows you the luminescence. If suppose if the primary antibody you used is a radioactive one, then you can go for X-ray film also, autoradiography machine. Otherwise, if it is chemiluminescent, then it is exposed to UV light, so that you can see the chemiluminescence, and then manual data assessment using the comparison of the gel and the membrane is done, and then you can tell that whether the protein you used belongs to a particular family or not, or it can be detected in that sample. So there are various applications of this method. Let's see the application. It is used for detection of particular proteins from a mixture of protein. You can check whether a protein is produced in an organism or not. Then size and amount of estimated protein in the mixture is also done. Verification of following high sensitivity ELISA test for diagnosis of Lyme, HIV infection, BSE, HBV, so on can be done using this. We can detect condensed isoform of proteins as well as tagged proteins also in some organisms. Then there are different advantages. It's an effective early tool diagnosis. Then detects minimal immunogenic response for viruses as well as bacteria, and it requires fewer antibodies for testing also, so it can be done for a multiple number of times, and it detects the protein from a large mixture of different proteins. Even more than 3 lakh proteins can also be there, and from that also you can detect a single protein. Okay, there are disadvantages also. It requires specific primary antibodies to perform the test on the desired protein of interest, means whatever the protein you are detecting should have a very specific primary antibody. Then it is challenging and hence required well trained staff, because the handling of this technique is a very important task. Poorer result, as the antibody may reveal off target binding. Suppose if you are not doing the wash properly, what can happen? The antibody can bind at others position and show a faulty result. Then detection and imaging, the results can be expensive as the equipment cost is high, because the autoradiography machine or the gel detection machine is a high cost machine. Thank you for watching. If you like the video, please press like button, share, comment and subscribe the channel. Thanks for watching.